function. Skeletal muscle, you know, is attached to the bone or the skeleton primarily. That's really where it gets its name. It also is attached to the eyeballs, so it moves the eye, but the rest of it is attached to the skeleton, to the bone. And it moves us, right? And it's very important. All of our movement that we want to do for that is the skeletal muscle. The cardiac muscle is the muscle in the heart. So that's going to pump the blood. That's what it's going to move. And then the smooth muscle is a little bit more varied. It's in the linings of most of the walls of most of our ducts, like the digestive tract and the respiratory tract and the urinary tract. It's in the lining of the uterus and the urinary bladder and all of these structures. So we have a lot of smooth muscle that moves things through ducts, and that's a very important function. We also have smooth muscle in the walls of many of our blood vessels, and they help with vasoconstriction versus vasodilation. So that's another place that we have smooth muscle. So those are the main differences in functionality location of those three types. But they have lots of other differences. This is skeletal, cardiac, and smooth. Skeletal muscle we're going to spend the most time with because it has a lot of features to look at. Cardiac will spend a fair amount, and then smooth, it won't take us too long to look at smooth. When we look at these three, first thing we can notice is, for odd reasons, we change the names of a lot of the cell structures when we're talking about muscle. That's odd, isn't it? And so instead of the plasma membrane, we can call it the sarcolemma. Instead of the endoplasmic reticulum, we can call it the sarcoplasmic reticulum. Instead of the cytoplasm, we can call it the sarcoplasm, for instance. And so we use this term sarco, and then we also use the term myo when we're talking about muscle in many ways. And so hopefully that won't disturb us too much that we have some of these specialized features, specialized nomenclature, naming. When we look at muscle tissue, because the muscle cells are so long compared to most cells, we often use the term fiber synonymously with muscle cell. If you say there's a skeletal muscle fiber, you can very legitimately be calling that one cell. It's a fiber. Now that's a little confusing because in histology we say we have specific terms for fibers. We have collagen fibers, and we have elastin fibers, and we have reticular fibers. These are protein chains, not cells. But in muscles, we sort of change the rules a little bit. You can call a muscle cell, the entire cell, a fiber. And it kind of gets from the fact that it's so long that it looks kind of like a fiber in that sense. So that's a little bit different as well. I'll go ahead and put a little pointer here that's something I do. So that's what we're looking at. Now within the muscle cells, of course, we do have a lot of proteins. It is the interaction of the proteins which really causes the contraction and the force to be generated. And a lot of these proteins, they have a specific name, but they can also be called myofilaments. Those are the smaller diameter one. The myofilaments are actin, a bunch of actin proteins put together. And then we have thicker filaments, and those are called myosin. When we say thicker, that just means they're bigger in diameter. So myosin are bigger in diameter than actin. And then we're going to have some regulatory proteins that we'll talk about, like tropomyosin and troponin. So we have a number of proteins in the muscle cells that do different things. All almost all cells, almost all muscle comes from mesoderm, the embryonic connective tissue mesoderm. Mesenchymal cells differentiate into muscle. The only exception is the iris, the colored part of our eye that is an aperture for the pupil. That is smooth muscle, but it comes from ectoderm, and it has different characteristics than all the other smooth muscle. So there's a little bit difference there. Here's the different types. We know, you probably know, that skeletal muscle are voluntarily controlled. Skeletal muscle are striated. It means they have light and dark bands, for instance. Cardiac muscles are striated, meaning they have light and dark bands, but they're involuntary. And smooth muscle are involuntary, and they don't have any striations. That's why they're called smooth. They're not banded. They don't have light and dark bands. So those are fairly common differences between the types, but there's a number of other issues that they have some differences that we'll look at. How are they made? Again, they all come from mesoderm essentially, and they form little structures called myoblasts and then myofilaments, and finally they make the muscle cells. This diagram contains the major differences between our three types. Here's skeletal muscle in this column, and here's cardiac muscle, and here's smooth muscle. So you can see the cells. By far the longest of the cells are the skeletal muscle, then the cardiac muscle, and the shortest are the smooth muscle. Skeletal muscle are unbranched, cardiac are branched. Now, how many nuclei do we have per cell? Skeletal, we have many. There's many, they're multinucleated. Cardiac, one or two, and they're centrally located. In skeletal muscle, if you think of a cylinder, like a paper towel, cardboard, it's a cylinder shape, right? The skeletal muscles kind of look like a cylinder, and all of their nuclei are on the outer edge, right underneath the sarcolemma, the plasma membrane. We sometimes call that hypolemally. It just means it's right under the membrane. In other words, they're not in the center of the cylinder, they're on the edge of the cylinder. Sometimes they'll look like they're in the center, but that's just because if you happen to catch a nucleus and you've got the cylinder edge and you happen to catch it right where it's next to that membrane, it might look like it's in the center, but they're all on the edge. And so that's interesting. Whereas cardiac muscle, this does this sometimes too, for some reason it pops off. Cardiac muscle, they are usually in the center, and smooth muscle, the nuclei are in the center. In cardiac muscle, one or at the most two nuclei per cell, smooth muscle, one. If you have actin to myosin, thin to thick, skeletal has 6 actin for every 1 myosin, or thin to thick. And so does cardiac, but smooth has 12 to 1. We'll talk about transverse tubules, T-tubules, but they occur at the AI band in the sarcomere, and we form triads, whereas at cardiac they form at the Z lines and they form dyads, and there is no T-tubules in smooth muscle. Some of these other things we'll pick up as we go through. One other thing to mention, cardiac muscle has intercalated disc, and we'll go over those. I think you've probably heard of those, and we'll look at those in the microscope. We'll see them. And then, of course, here, skeletal is voluntary, the other two are involuntarily controlled. So there's some of the major features. We're going to focus in on skeletal muscle for a while. This is examples of skeletal muscle. And this is only 160. Let me crank this down again. Now that we're looking at slides, we'll make it darker. And here you can see the nuclei, and again, the nuclei are on the edge. You can have some connective tissue in between these skeletal muscle fibers. So like even here where you're seeing a little pink, like it's teased apart, that's skeletal muscle that's connective tissue around the skeletal muscle. But these are skeletal muscle fibers, they're sort of running up and down. If you look carefully right here at our 360 magnification, you can see the striations pretty clearly. At this magnification, it's a little harder to see them, but here and here, you can see the striations. And as you look for them, the striations are pretty easily defined there. Now, this is a beautiful slide, isn't it? The fibers or skeletal muscles are running in this direction, like if it's from our left to right as we're looking at it. And you can see the striations, see the darker, lighter, that banded pattern. And sometimes this is described as stacking logs on top of each other. That's kind of what skeletal muscle fibers look like a little bit. And notice that the nuclei are at the edge. These are the nuclei, they're pretty flat. What about these? Well, these are at the edge too, but they're around the outer edge of the cylinder, but they happen to be right at that part of the cylinder. So don't be fooled that they're in the middle there, they're on the outer edge of the cylinder. These, of course, look very obviously to the outer edge, but these are as well, it's just the outer edge of that cylinder. This is a little bit of connective tissue. We have a special name for the connective tissue and muscle. The connective tissue around each individual muscle is called the endomysium, and around a bundle of muscles, a fascicle, it's called the perimysium, and around the whole muscle, it's called the epimysium. We're going to show you that in the slides later, eventually, so don't worry if we don't get that right now. But this is connective tissue, which would be endomysium, and it helps hold things together there. So this is skeletal muscle. Here it is, another beautiful slide. This is really showing, we can even see the subunits called the sarcomeres. This would be one Z line here to another Z line over here. From here to here is a sarcomere. So we have all of these microscopic subunits in skeletal muscle called sarcomeres. We're going to get well acquainted with the sarcomeres, and we can see them really well here. And you can certainly see the banded, darker, lighter banded patterns here. Again, this is a muscle cell or fiber, and here's another one, and here's another one, and they're very long. These are the nuclei at the edge, skeletal muscle nuclei. This is actually a nucleus right here of a fibroblast. It's outside of the skeletal muscle fiber. You see the little connective tissue here? This is a fibroblast laying down, of course, connective tissue in between the muscle fibers. This is probably the same thing, a fibroblast. But these that are actually in the muscle cell are the nuclei of the muscle cell. Yeah.
So from the little dark, light, dark bands,
Right.
That's the sarcomere?
You go from one, see this real little line?
Okay, yeah.
That's a Z line.
Okay.
And you go to this Z line, so from here over to here, that's a sarcomere.
Right. So
This is gonna be what we call the A band, right in here, that darker region. So yeah.
okay.
And here is some sarcomeres. This is an electron micrograph. So here's a nucleus. And in this case, they're going up and down. So this is one I band, or Z line, excuse me, and then to another I band. So the sarcomere is going from here to here. This would be the A band, there's the H zone, and this is the I band where it's lighter. So we're gonna learn those different regions. Hopefully they have some familiarity to you, but we'll learn them, so don't worry if you don't. But these are the sarcomeres. These are these small little subunits in the muscle cell.
So on the previous slide, on this one, so it's not that each one of these fact-being, like structures are a fiber, like the fibers are in them, so this whole thing is one fiber.
This whole thing is one cell. We could also call it a fiber. Okay, so we have a cell, and then it has a bunch of proteins in it. These proteins are arranged in some orderly fashion that make up the sarcomeres. Those proteins are mainly myosin and actin. And the arrangement of the myosin and actin gives us these sarcomere subunits within one muscle cell or fiber.
So this is one muscle cell.
That's one muscle
and then
cell.
each of these going this way is the,
Except they're going this way and that. They're going left to right in that way.
oh, but,
They're not going between your red lines, they're going from here to here.
so.
This is lengthwise, going left to right. This is this cylinder going up and down vertically. Oh, you have it right. From there to there is this, from there to there is a sarcomere. What I thought you were saying was, from here to here. It's from here to here.
Oh.
So this is one myofilament proteins. So you have several myofilaments in one muscle cell. Confusing, isn't it? A bunch of myofilaments, some of which are thick myosin, some of which are thin, are inside of this cylinder of the cell muscle or fiber. And those are arranged in ways that make these sarcomeres. So in that slide you're looking at, let's go back, they're arranged from left to right. Here's one sarcomere, here's another. In this slide, they're arranged vertically. It's oriented differently. So it's like from here to here's a sarcomere. So if we took this and we flipped it sideways, it would look the same as this. But of course, we're seeing more detail here because this is an electron micrograph and this is a light micrograph.
Were you saying something about how there's different parts in one of the cells like this way, or you were only saying there's different parts this way?
Sorry, say that again?
Were you only saying there's different parts like this way, like the myosin and the microfilaments, or did you also say it's like different this way?
No.
No?
It's
Okay,
just-
this way.
In one, we're looking at the cell looking like this. And the next one, we're looking at the cell looking like this.
Okay, but in this picture where it's like this, you weren't saying that there's different parts in the cell this way, only this way.
Only left to right.
Okay, got it,
That's
thank you.
Yeah. That's good to visualize it right like you're doing because that helps us understand, you know, when we can visualize it, it makes it make more sense. Now, what are the proteins in the skeletal muscle? They are the thin filaments, microfilaments or actin, and the thick filaments, myosin. But then we also have some regulatory proteins, tropomyosin, that sits on top of the actin, and troponin, which sits on top of the tropomyosin. And there's three subunits of troponin. Troponin T, that attaches to the tropomyosin, troponin C, that calcium binds to, and troponin I, which inhibits the interaction between the thick and thin filaments, the myosin and the actin. So these two, tropomyosin and troponin, are regulatory proteins. By the way, muscle cells, when they reach maturity, they cannot divide. If you take a chunk out of your skeletal muscle, it doesn't divide. What if you go to the weight room and you're working out? The muscles you currently have get bigger. You add more protein to them and they get bigger. You don't add new muscle cells, right? You don't add new muscle cells once you reach maturity. You add size to your current muscle cells, your already existing muscle cells. That's the same for cardiac muscle. It doesn't regenerate. Smooth muscle can regenerate. It can, to a certain degree. It can undergo mitosis, but not skeletal and not cardiac. So there's a little bit of difference there. Now, when we look at the actin and myosin. We have the thick filament and the thin filament. Here's the actin in green. There's the actin. And on top of the actin, you have tropomyosin. That's this one here. And on top of that, you have this globular troponin with the three parts. Troponin I, troponin T, and troponin C. What happens is when you want contraction, you want the myosin to bind to the actin and to pull the actin to generate force, but the myosin cannot bind to the actin if the tropomyosin and troponin is blocking that interaction. To disinhibit it, we have to bind calcium to the troponin C part of the troponin, and then it pulls the troponin, pulls the tropomyosin out of the way. It slides it out of the way. And the cross bridge of the myosin can connect to the actin and initiate contraction. And that's how we're going to initiate muscle contraction. Here's the myosin with the cross bridge, or the head, as it's called. This is the part that wants to bind to the actin. And so it wants to bind, but it can only bind if the tropomyosin troponin is temporarily slid away out of the way. When we're looking at the myosin and the actin, we have this sarcomere. This part is myosin, or thick protein. This part is the actin, or the thin protein. And they're arranged in this kind of a pattern that we call a sarcomere. From your book, you can see a diagram of the sarcomere. Again, this thicker green line, that's the myosin, and these thinner green lines, that's the actin. So they're arranged in this pattern. At rest, a sarcomere is from one Z line, one here, to the next Z line. So there's a sarcomere. Where you have myosin from here to here, here to here, that's called the A band. Where you have just myosin, no actin, see how the actin and myosin are both here, but where it's just myosin, you have the H band. And then very middle is called the M line. When you have contraction, the actin actually slides towards the center. So you reduce your H band, and your actin slides towards the M line. And so that is in the contracted state. This is in the relaxed state. So we want to know the different parts of the sarcomere. Here's the sarcomeres in an electron micrograph that we can actually see them. Here's one Z line to another Z line. So this would be the I band, this would be the A band. If we look carefully there, that lighter part right there, that's the H zone. And so those are the different parts. Here it is where we can see it a little more easily. We've blown it up, made it a little bit bigger. This is the Z line right here. This is the other Z line. So from one Z line to the next Z line, that's one sarcomere. The I band is where you have only actin from adjacent sarcomeres, like from here and here. So this is the I band, this lighter staining region. This is the A band. You can see it on the list here, but it's from here to here. Here is the H zone, right from about there to there. And then there's the M line right in the middle. Here's the other Z line. These are all mitochondria, by the way. This and this and this and this and this and this. Remember, skeletal muscle's got a lot of mitochondria because it's very energetic. Makes a lot of ATP. This is looking sideways at the sarcomere. So if you took this and you turn it sideways, the thicker, that's the thicker myosin proteins, and these smaller dots, that's the actin. So this is myosin, and this is actin, looking at it in cross section, looking at it sideways.
Can you say that again?
So if you took this and you flipped it sideways, so instead of looking at it lengthwise, you're looking at it
Okay.
at the cross section. You're looking at it in there. So all the myosin and actin are running along lengthwise, but now you're just seeing the ends of them. So they look like points. Well, one point is a little bigger around than the other because myosin is thicker and actin is thinner. So it's like looking at a thicker pencil and a thinner pencil. So what we're looking at is the bigger, darker ones are myosin, like here and here and here and here. And the thinner ones, the little ones, that's actin. And so that's looking at them from the end sort of perspective.
But they're both the black holes, but it's just whether it's the big one or the small one?
That's right.
Okay.
The big dots are the myosin, the little dots are the actin.
Perfect.
Yeah. This shows it with a diagram. Here's your myosin with the cross bridges or the heads. Here's your actin. Here's your Z line. There's your H zone and your M line. So that's what they look like. Again, the myofibrils are your actin and myosin in that pattern. And there's your bands, your I bands, your A bands, your H zone, your M line. Now, around inside of your muscle fibers, you also have, here's your myofibrils, that's your actin and myosin. You have some sarcoplasmic reticulum. Now, the sarcoplasmic reticulum is an endoplasmic reticulum. It's really what we're focusing on here is the smooth ER and the ones that's not studded with ribosomes. And the smooth ER, well, it's the sarcoplasmic reticulum in a muscle cell. It has a special purpose and function. The sarcoplasmic reticulum in a muscle cell has membranes around it, and it has calcium ions sequestered inside of it. They've been pumped in there, active transport. And they pump it to a specific part called the lateral sacs, the edges of the sarcoplasmic reticulum. And so we have calcium inside of the sarcoplasmic reticulum, which is inside of the muscle cell. What happens is when we generate action potentials along the sarcolemma, when we do what? When we generate action potentials along the outer plasma membrane, the sarcolemma of the muscle cell, it runs along the length of the muscle cell, but it goes into the cylinder using transverse tubules. Transverse tubules are like a membrane that goes inside of the cell. So here's the sarcolemma, and here's a transverse tubule. They're called transverse because they're running transverse or perpendicular to the sarcolemma. And where does that action potential going along the transverse T-tubules go? Sarcoplasmic reticulum. Specifically the lateral sac or the edges of the sarcoplasmic reticulum, and that causes calcium gates to open in the sarcoplasmic reticular membrane. Calcium goes out of the sarcoplasmic reticulum into the sarcoplasm, the cytoplasm. And that calcium can then bind to troponin C, which temporarily changes its shape and slides troponin and tropomyosin out of the way, so myosin can bind to the actin, and we can initiate muscle contraction. And so that's how it happens.
Is the terminal cisternate, is that part of
That
the
is,
sarcoplasmic?
That's the lateral sacs. Yeah.
Okay.
The cisternate just means it's a little bit of a swollen area of the sarcoplasmic reticulum. And it's also called the lateral sacs.
Okay.
So here's the path of the action potential. It comes down the transverse tubules. and it gets to these lateral sacs or terminal cisternae, and it allows the calcium to come out. So that's exactly right. And then it allows the interaction of myosin and actin. This is showing mitochondria here too. Okay. Now, in skeletal muscle cells, the transverse tubules come down between the A and the I bands of the sarcomere. In other words, here's the A band, here's the I band. They come here. The reason that we want you to notice that is in cardiac, they come at the Z lines. They come in a different place. But here they come for skeletal muscle, these transverse tubules come to this area between the A band and the I band. So that's the A band, A-I band junction, and that's where they come through. It's called a triad because you have a lateral sacs or terminal cisternae on each side of the transverse tubule. So you have a transverse tubule and a lateral sac here and a lateral sac there, so it's 3 parts, and that makes it a triad. In cardiac muscle, it's only a dyad. There's a transverse tubule and 1 terminal cisternae or lateral sac. So it looks a little different. This is your motor end plate with your motor neuron going down to the skeletal muscle and releasing acetylcholine. You may remember from Systems Phys, and you may well not, that, by the way, your wall is down there. I just noticed that. When you have a motor neuron going down to the skeletal muscle, it always releases acetylcholine, and it always binds to a nicotinic-cholinergic receptor, and it's always excitatory. We're not going to be getting into physiology too much, but that's just to sort of link it in to some things that you maybe already know or maybe kind of remember a little bit from physiology. So at this linkage point where we're linking it, we're releasing acetylcholine at this little synapse between the motor neuron and the muscle, and it's going to go bind to nicotinic cholinergic receptors. So over here we have the nerve, and over here we have the muscle, and here's the little synapse right here. Sometimes it's called a myoneural junction or a motor end plate, but it's just from nerve to muscle. So that's what's going to happen. Again, we're going to release acetylcholine, and it's going to bind and cause this contraction. Again, yeah, we're okay on time. When the muscle is relaxed, the tropomyosin is blocking the head or the cross bridge of the myosin from binding to the actin. And on top of the tropomyosin is troponin. When you have the action potential and you release calcium that binds to the troponin, it slides tropomyosin out of the way. The cross bridge binds to the actin and it slides it in towards the center of the sarcomere. And that's going to decrease the size temporarily of the H zone.
Does it also decrease the size of the I band?
The I bands are pulled together.
Okay.
So the whole sarcomere would be a little bit compressed. Because it's sliding one over the other, it's, you have more, the myosin and actin are now more overlapping than they were. Now, ATP is needed and for this energetic step, and it's also needed to unhook the myosin cross bridge from the actin. And so that's also an energetic step. So you have to have ATP for that. And that's why you remember that after death, you can get rigor mortis, rigor after death, because you don't have enough ATP to disengage any muscles that were contracted during death. And And so after a while, those proteins degenerate. But for a while, like if somebody died and they were sitting up against a wall, well, you couldn't lay them down flat for a while easily because those muscles are still in their engaged, contracted scenario. And you don't have enough ATP no ATP is being made. Now, when you pump the calcium back in to the sarcoplasmic reticulum, then it leaves the troponin and it goes back into a relaxed state. We get energy for ATP from glucose and fatty acids. And here this shows you the cross bridge binding, and you need ATP, and then you also need ATP for it to disengage. Here's your motor neuron coming to your skeletal muscle, and then it goes action potentials down the length of the sarcolemma, and then into the interior of the cylinder through the transverse tubules to the lateral sacs of the sarcoplasmic reticulum. Calcium is released through channel proteins called rhyodine receptors, you may remember. Now this is from your physiology book, so sorry to give you nightmares. You're not needing to memorize all this again, but if you wanted to review all the steps, this shows you the major steps, physiological steps of muscle contraction. And that's just to, if you're wanting to correlate the histological anatomical events with the physiological events, that just shows it there for you. But we're not going to ask you like an essay or anything, so don't worry. That's just for your sort of to reference it back to the physiology. We briefly mentioned that skeletal muscles, we have connective tissue associated with it. The connective tissue that's around an entire muscle, like the biceps brachii, for instance, is called the epimysium. We have bundles of nerve cell fibers called fascicles, and each fascicle is surrounded by connective tissue called perimysium. And then around each individual muscle fiber, we have connective tissue called endomysium. So if we're looking at the entire muscle, connective tissue around it is called epimysium. If we're looking at these bundles of fibers called fascicles, the bundles of fibers, then the connective tissue around there is called the perimysium. And around each individual muscle fiber, it's called the endomysium. So we'll be looking at that as well. And of course, muscles have lots of blood vessels associated, and they run through the connective tissue. It's the same with nerve. This is a cross-section of skeletal muscle. So we're looking at it in cross-section. So if you have a bunch of muscle fibers running longitudinally, if we're looking at the edge, they're going to look like this, right? We're going to look at them from here. Now the nice thing is, if we see skeletal muscle fibers in cross-section, we can really see that the nuclei are only around the edge, not in the center of the cylinder. And so that really helps us identify it as skeletal muscle. Look at this. These are in cross-section. This is a skeletal muscle fiber. Look at the nuclei. Where are they? They're at the edge. Here's another one. The nuclei are at the edge. And we have more than one. It's not just one nucleus. We may not see, it's just at the end, but here's another one where there's the nuclei at the edge. We don't see one in the center. We don't see a nucleus in the center. This is connective tissue. If you have connective tissue around an individual muscle cell, it's called endomysium. This connective tissue would be endomysium. If you have one around a whole bundle, it's called perimysium. What do we have here in the connective tissue? Well, this is an arteriole. This is a blood vessel in cross-section. This is a vein. We're gonna look at those a little later on in this quarter, right? But that's what these are, if you're wondering. So this is connective tissue, this is blood vessels, and these are the skeletal muscle cells, and this is the connective tissue around it. So that's what we're seeing there. This is with a special stain. This would be, each one is a skeletal muscle cell here. So around each one, this would be endomysium, and then the connective tissue around a fascicle here, that would be paramysium. This is a blood vessel, so it doesn't pick up that stain very well, but a little bit. All this connective tissue from the epimysium merges together and forms tendons and ligaments. From muscle to bone, it's a tendon. From bone to bone, it's a ligament. Both connective tissue. So in this case, it would be a tendon. All that connective tissue joins together. How would we classify it? Dense, regular connective tissue, right? Made out of collagen. That's what it would be. That's just connective tissue that connects the muscle to the bone. And it's our tendons. And that's how it does it. Okay, we're already past time. Wow. How the time flies. Okay, so tomorrow at 2 o'clock, we meet in Clark Cultural. We'll go a little bit more over the muscle. We won't get too carried away. And then we'll go down to the lab room and we'll look at the blood